Bismillahirrahmanirrahim. Yes, my dears, third and last portion of the bacterial cell is the cytoplasm. And we studied that cytoplasm will be consisting of cytosol, which is the soluble part of the cytoplasm, and internal structures. This cytosol may contain different things, different ions and molecules inside it. And as far as cell structures are concerned, they are included the nucleid, plasmids, ribosomes and other inclusions, जिनमें हम बात करेंगे गैस वैक्यूल्स की, मैग्नेटोसोम्स की और साइटोस्केलेटन की. So first of all let's see what is the cytoplasm and how it is present inside the membrane. Inside the membrane in bacterial cell is the cytoplasm. Membrane is actually wrapping the cytoplasm, and cytoplasm is a semi-fluid mass of proteins, amino acids, sugars, nucleotides, salts, vitamins and ions, etc., which are dissolved in water. And inside the cytoplasm there are several subcompartments which have some specific functions, and these subcompartments include nuclei. First of all, nuclei. I've told you, this suffix oid, when it comes to any word, it means actually like the previous word. Nucleoid means just like the nucleus. It is not nucleus because there is no membrane around it, but it serves the function of the nucleus because it contains DNA. But there is no ribosome in prokaryotic nucleoid region, and there is no nuclear envelope. There is no histone proteins which combine with the DNA molecule to form chromosome. So although some of the biologists they call it a chromosome. Suppose this is a bacillus bacterium, and inside it there is a region which contains its chromosome. And it is not actually the chromosome because it does not contain protein. It is just the DNA molecule, and it is a single structure, not in a diploid form. So most of the prokaryotic cells are haploid cells. So chromosome is not present. As far as genome of bacterial cell is concerned, it consists of few of the genes. For example, E. coli, which is the mostly studied bacterium, its genome consists of 4300 genes only, which can translate 2000 different proteins. So nucleoid is the central and very important subcompartment of the bacterial cell, which is contained DNA, which will control all the metabolic reactions of the cell. Plasmids are some extra chromosomal DNA. There may be one or more plasmids in a cell. For example, this is the chromosome, and these are the circular DNA molecule which are extra to the main genome of the cell. So these are plasmids. They are named as plasmids. They contain 5 to 100 genes, and there may be one or more plasmids. They are not needed for growth or metabolism of the cell, but they provide genetic flexibility to the cell. And plasmids contain some disease resistance genes, or they may contain some toxin genes against the toxins, which can produce protein against the toxins. So that's why, because they are containing the resistance genes on them, so they are also named as R plasmids. R stands for resistance plasmids. Then are the ribosomes. Inside the bacterial cell, there are hundreds and thousands of ribosomes, and there's so much abundance in the cytoplasm actually make the cytoplasm a granular structure. And they are 70 S ribosomes. In eukaryotic cell there are 80 S ribosomes. Here their smaller subunit is 30 S and larger subunit is 50 S. S stands for Swedberg unit. It is the unit for centrifugation. So there are some antibiotics. When we use antibiotics, against any infection. What do the antibiotics to the bacterial cell? Some of the antibiotics they inhibit or they block the ribosome to produce some specific protein. And when bacterial cell cannot produce their specific proteins, they are killed or they cease to grow. So that's why we get rid of the diseases. Other than ribosomes, there are some other inclusions bodies which store different nutrients. For example, some of the inclusion bodies are there which store glycogen inside them, and there are some others. For example, diphtheria bacilli contain some metachromatic granules or volutin, which deposits polyphosphates. So there are different inclusion bodies inside the cytoplasm in different bacteria, and they store different types of nutrients. For example, some of them are storing in E. coli, they store glycogen and some other bacteria, but in diphtheria bacilli, which causes dysentery, that contain polyphosphates in its inclusion bodies. Then there are uh, some in case of some aquatic prokaryotes, there are some gas vacuoles which provide buoyancy to the cell, and there are magnetosomes. Some magnetosomes are there in some of the bacterial cell. Magnetosomes are what? What are magnetosomes and what are gas vacuoles? Where gas vacuoles are present in aquatic prokaryotes, they are most oftenly present in cyanobacteria, but there are some bacteria which also contain gas vacuoles. They provide buoyancy so that they can swim in water. They do not let the cell to sink on the bottom. Magnetosomes are the invaginations of the cell membrane. These magnetosomes provide, they increase the surface area for the enzyme to react inside the cell, because all the metabolism is to take place inside the cytoplasm. There are no other cell organelles. So these membrane invagination provide the site for many enzymes to work. For example, ATP synthesis also takes place on magnetosomes, some kind of magnetosomes, and there are some magnetosomes which form the mesosomes, which help in the cell division, जो कि cell division में help करते हैं. In the end, I would let you know that there is cytoskeleton as well. It's very astonishing that prokaryotic cells have cytoskeleton. Up till a few years back, it was considered that prokaryotic cells do not have any cytoskeleton. But recently, it has been discovered that there are some homologues of tubulin and actin protein within the cytoplasm of the bacterial cell, like E. coli and other bacilli as well. They have some tubulin and actin homologue. Homologue means they have the same attributes as that of the tubulin and actin protein, and they combine together to form some microtubules. Although they do not form microfilaments, but they aggregate especially during the cell division, during the binary fission, and they help the cell in maintaining the structure and giving the support to the cell, as well as they also help in the binary fission. So cytoskeleton is also present in most of the prokaryotic cells, but that is not exactly in that condition which is found in the as the cytoskeleton of the eukaryotic cell. There are some protein molecules which can combine together. They can assemble and disassemble, just like the tubulin and actin protein of eukaryotic cell. So all this is about the general structure of a bacterial cell. I hope you understand the general structure of bacterial cell in more detail as compared to your previous knowledge. Anyhow, if you have any question, you can ask me in the WhatsApp. I will try to answer your question. As far as more detail of these structures, for example, we can prolong the lesson of nucleoid and plasmids. Plasmids are very, very important. They are being used in industrial biotechnology, genetic engineering and biotechnology, gene manipulation. They are now being used. They are now an industrial product. These are the plasmids which are helpful in the formation of the insulin, which is being commercially prepared now with the help of bacteria. So, this was today's lesson. If you have any question, you are most welcome. Thank you very much. Allah Hafiz.